Now there are again a lot of topics that I need to go through today, so I'm only going to summarize the main things I want you to take away from this chapter. So the first one is actually talking about cell division. Before we talk about this cell division, we need to take note of the structural features, okay, that is related to mitosis and meiosis. So remember, there's always key phrase to actually describe. So if they ever test you something like, for example, okay, your centrioles. What are always the two ways you describe your centrioles? Can you tell me first, Hannah? Whenever you want to describe centrioles, like for example, the typical kind of questions they like to ask, what is this structural feature? Then later they say, give evidence from your diagram to show that this is actually a centriole and this is typically 2 marks. So what's the first thing that we always write for your centrioles description? It's always a pair of what? A pair of what? Please know the phrasing, Cheryl. A pair of? No.
No.
What is this two light called?
Sorry. Rod light.
Continue. Haven't finished yet. Rod-like structures, and then they are always perpendicular to each other. Okay. Next one. What's the second phrase that we always describe, dude? Ethan?
9 triplet
Of
microtubules.
Arranged in a
Cylindrical.
ring. So 9 triplets of microtubules arranged in a ring. Okay, so please actually write down if you actually don't see that in your notes. Okay, my triplets of microtubules arranged in a ring. So these are always the two description if you ever want to describe your centrioles. Understand? Now another thing we also need to take note is your centromeres. So you notice how your centromeres is a region here. Centromeres, they are associated with 2 types of proteins. First type of protein is called what? Can you tell me, dear? Cohesion. Huh? What is this that I'm pointing?
Oh, the
Start K. Please know. Is your kinetochore protein. So your centromeres, what some features your centromeres have so you can attach to kinetochore proteins? It must be, what's always the thing so that the kinetochore proteins can attach? Must be
complementary.
So this is the usual phrase you always see in exam if you want to associate with this, because you already should know cell division, mutation, cancer, and even cell signaling sometimes can actually go hand in hand. Alright, so please know things like complementary in shape is going to be the standard key thing, but let's discuss. Usually, this is an exam. Whenever you want to say how your centromeres, number one, main function is they associate with kinetochore proteins, right? So what is the purpose? Can you help me tell me how do you usually write an exam? So when your centromeres, first function of centromeres, they associate with kinetochore proteins, because if you just write like that, I cannot give you 1 mark at all. So what must we write? Hannah? For what?
Separation.
What is this?
Spindle fibers.
To
attach.
Yes. I mentioned this time and time and again already. If students just tell me centromeres just associate with kinetochore proteins without telling me for spindle fibers, okay, to attach, no marks, alright, at all. So you must already always say it's for spindle fibers to attach, it's a compulsory thing. The question is, there are 2 reasons why your kinetochore proteins must allow spindle fiber to attach. What's the first reason? Can you tell me, Cheryl? The way we always write for exam. First reason is what? To ensure, like in metaphase, keyword is what? Sorry?
Is it the equal separation?
No.
No.
Is what? During your metaphase plate?
The
Keyword is?
the proper alignment
Of
of
chromosomes along the equator. Keyword is proper alignment of chromosomes along the equator. And you will realize that usually alignment, proper alignment are always the standard key phrase you want to describe. Second thing is what? Why do we also need to associate with kinetochore proteins, Yunchin? Why must we associate with kinetochore proteins? To ensure what? Equal separation of what? Equal separation of your sister chromatids, alright? Or your chromosomes. Keyword is equal separation. Okay. Sorry, I forgot to write equal. So equal separation of sister chromatids of chromosomes alright, to opposite poles of the cell. Cannot write opposite sides, you always have to write opposite poles because it's north pole, south pole. Alright, so take note about the keywords. Now, because we have your centromeres associated with kinetochore protein, centromeres don't only associate with kinetochore protein but another set of protein. What is that another set of protein called? Any idea? Ethan? Start with C. Co.
Cool.
You know? Cohesine proteins. So again, this is another set of proteins I want you to take note of. So again, they associate with cohesine proteins. The purpose of associating with cohesine proteins is for your sister chromatids to anneal to each other. So, what is the purpose of this cohesine proteins here? Same thing. Number one, if your this cohesine proteins doesn't allow your sister chromatids to adhere, you will not allow proper alignment. So they have the same exact thing. So first one is that you ensure proper alignment of sister chromatids along your this equator here. Second thing is, during your anaphase, cohesine proteins must be cleaved, must be degraded. So if they are actually degraded, it ensures equal separation of sister chromatids. Do you know why we need to know all the different functions of these proteins here? Because as I already said, it's related to mutation, right? The type of questions they ask, they can sound tough. Like for example, how come we can have non-disjunction? How come we can have aneuploidy? Reason is because sometimes you can say, okay, there can be a mutation in your cohesine proteins such that it cannot be degraded. So that's why you can have non-disjunction. Or you have mutation in your centromeres, which doesn't allow your this cohesine proteins or your kinetochore proteins to attach, so no longer complementary in shape. So that's why they cannot allow for equal separation, so you have non-disjunction as well. So that's why we need to learn your these functions, okay, of your centromere well. Understand? Now, I'm not going to go through every single part of your this cell division. I'm only going to go through two parts. First part of your cell division is your checkpoints. Everybody should know your checkpoints are G1, G2, as well as M checkpoint. G1 and G2 are part of which phase? Can you tell me, Jude? G1, G2, part of which phase? In the,
What phase?
What phase is it?
S phase.
S phase. S phase is S phase.
G1 and G2 phase.
Part. Never mind. Interface. So it's part of interface, dude. So it's on your first page. If you're not sure. Okay. So we have this interface. So with G1, S, and G2. Now, in exams, they can actually, I've seen this in essays before. Alright. Please know what do G1, G2, and checkpoints actually does. Alright. So if you haven't done so, maybe you can find some space to actually write at the side. For your G1, let's try. Can you tell me, Ethan, what's the first row for G1 checkpoint 4? Is to ensure?
DNA damage.
Oh, yes, again, so you want to check for absence of DNA damage because you should know right after your G1 is actually your S phase. So when I say DNA damage, that means you also want to check whether there is sufficient organelles, alright, or even your this adequate cell size. So I'll just write down. Sufficient. Okay, organelles. That means you say duplication of organelles. Let me write sufficient or duplication. Duplication of organelles. Okay, or you can even tell me adequate cell size. So you want to check for all this. But the second row for your this G1 is what, Ashley? What's the second row of having your G1 phase? Second is, like for example, what will stimulate your this cell division? Must have presence of. What is the ligand that will stimulate cell division? The ligand to stimulate your this conversion of excess glucose to glycogen is insulin. How about cell division? Is growth hormone, okay, or growth factors. So you will always want to check for presence of growth factors. Alright, So all this, right, must actually check before they proceed to the next phase. Understand? Now, your G2 is for what purpose? It's right after your this S phase. So obviously we know it's to make sure DNA or ensure DNA replication is complete. I'm just writing in case if they test for essay, so please know what is the purpose. So ensure DNA replication is complete. Okay, what else also are we ensuring? No DNA damage as well. M checkpoint stands for what? So M stands for metaphase. Purpose of M checkpoint we typically write is to ensure proper attachment of all chromosomes to kinetochore, okay, microtubules. So again, proper attachment is the keyword of all chromosomes to kinetochore microtubule. Also known as your bipolar tension, since they are going to align them along the equator. Okay, please specify what kind of microtubules you are checking for. Alright, because again, I already mentioned there is also polar microtubules, right? A quick one, can you tell me, Hannah, what is the function of the polar microtubules? Kinetochore microtubules is meant to attach to kinetochore protein. But we also have this thing called polar microtubules. What's the purpose of polar microtubules?
They lengthen
The cell, because you need to undergo anaphase, you need to undergo telophase cytokinesis, right? So your polar microtubules is to lengthen this cell. Understand? So ensure all chromosomes also are properly aligned. So do you see that's why the proper alignment is usually the generic keyword here? Okay? So all chromosomes properly aligned along metaphase plane. Okay, or along equator. Before dividing cell can enter this S phase. Now, however, I say, if let's say, right, you cannot remember their functions, alright, individually, collectively, what is the purpose of this checkpoints? Can you tell me, Cheryl? Remember I say, first one is PPP, stands for what? Prevent. So all this having these checkpoints is for prevent, progression of what? The cell cycle. So I repeat, first one you will always say is to prevent, the PPP, prevent premature progression of this cell cycle. The second one is actually for what? Any idea? Provide what? Provide time for what? For cell machinery to repair itself should there be any damage. So collectively, these checkpoints, okay, they have number one, prevent premature progression of the cell cycle, PPP. Second one, you always say is to provide sufficient time for cell machinery to repair itself should there be any damage. Okay? Now, the third thing I want to go through for this cell division, alright, is again, things that can actually increase this GV when that is only under this meiosis. In your notes, if you have here, because I'm not going to go through the step-by-step phases of cell cycle, I expect you to know. So in your notes, do you see we have this section called importance of this meiosis? Okay, so let's go through. There are two main importance of meiosis. The first one, I write down there sexual reproduction, but if you just receive the notes, can you write at the bottom? Standard key phrase you tend to write for the functions of meiosis is, ready? So again, it's here. If you don't know where I'm talking about, which is on the second last page of your notes, alright, this first column that you see sexual reproduction here. Alright, so right below the sexual reproduction mark, you will write is to form haploid gametes. I repeat, the first function of meiosis is to form haploid gametes in order to restore diploid condition upon fusion of gametes. Now, but the second one is the more important one. From now onwards, never ever just tell me you want to increase GV. Standard key phrase, right, you will see most of the time as an examiner, they will always not only just say increase GV, they elaborated slightly with effort in order to give you the certain 1 mark here. So that's why, right, what's the typical way when we say why meiosis can also increase GV? Like, what's the significance of increasing GV, Ethan?
New combination of values.
No, haven't yet. I'm not asking for crossing over or independent assortment yet. I'm saying, like, what's the significance of increasing GV?
For natural selection to add on.
So, to increase what? Chances of...
Surviving when the environment change.
No, the standard way, can you all please write, is to increase chances of survival in a species, especially during changes in environmental condition. So I repeat, to increase chances of survival in a species. Okay? Especially during changes of environmental condition for natural selection to operate on. I repeat, increase chances of survival in a species, especially during changes in environmental condition for natural selection to operate on. Now, we have 3 ways in this meiosis that it can increase GV. Let's see whether you recall. First one, Ethan, what's the first one that actually increased GV there?
Mutation
Oh no, I mean for meiosis.
or crossing over.
How do you write for exam? Cannot just write crossing over, always must write together.
Phase 1, crossing over of non-sister chromatids of homologous chromosomes.
To form, okay?
Then exchange equivalent amounts of alleles.
So that one, right, the exchange equivalent amounts of alleles is not usually the keyword. What he just mentioned is, the keyword is, alright, crossing over between non-sister chromatids of homologous chromosomes. Do you all see this heading here? Help me write at the side. This is during prophase 1. This is always worth 1 mark. Don't just tell me crossing over. I must see non-sister chromatids of homologous chromosomes during prophase 1. So this contributes to GV, but my question is, how does this increase GV? How do you write?
New combination of values.
Yes, so can you help me write aside? How does this actually increase GV? Is that they form new combinations of alleles. Do you see here under the list, second last page of your notes, 3rd column? Alright, so take note. So again, what do I mean? For example, this is under your in this inheritance part. You learn that crossing over is always at the chiasma. So if I were to write, this one is, for example, big A, big B, big A, big B. Okay, then we have here this small a, small b. Crossing over can only happen during linked genes, okay? So do you see, upon crossing over, you can actually form the gametes that is as such. So for example, let's say small a with your this big B, and then again, we can have your this small a, big A, sorry, wrong color, big A, and then your this small b, and then same thing, your this small a, small b. So do you see I formed by new combinations of alleles here? This is only for linked genes. Question, is it partial linkage or your this complete linkage? If you can have crossing over. Get ready, I want to know what's the evidence you look out for in exam so you know the question is testing you on partial linkage. So, is it for partial linkage or complete linkage?
Partial linkage.
Partial linkage, because complete linkage they are so close together such that, can they be separated from crossing over? Can never be separated from crossing over. How will I know if a question is under inheritance is testing me on partial linkage? Number 1, what's the number 1 factor as evidence you see and search for in exam? Parental. They always give you a table, right? Show you the offspring, right? Phenotypes are
like a lot more.
significantly larger in numbers. Second one, between the parental phenotypes and recombinant phenotypes, what do you look out for? Between the parental phenotypes is about 1:1. Within the recombinant phenotypes, also 1:1. So sometimes they will have a 1:1, 1:1 ratio. This is the second evidence. Third evidence you look out for is that you can calculate what? Something frequency? Non-frequency. Sometimes they can give you in terms of centimorgan. If they can give you centimorgan, that's straight away recombinant frequency units already, testing you on your this partial linkage. So again, do you see what's the way we write for exam to contribute to GV? This one is, how do you write for exam? Crossing. How does this, okay, how do you describe this process? of
between
Non-
sister chromatids.
non-sister chromatids of?
Homologous chromosomes.
Okay, so this during prophase? Okay, so this actually contributes to GV. How does this increase GV? They form new, keyword is what? You have to write new. Those who actually let me mark for your this practice paper, some words that you never write new, no marks at all. Okay, so take note about that. Second one, what's the second thing that contributes to GV?
Independent assortment of homologous chromosomes at metaphase 1.
So again, metaphase 1 alone is when you actually have different random arrangement of maternal and paternal genes or chromosomes facing one pole of your cell. So something like that. It doesn't mean all the maternal chromosome will face one side, all the paternal chromosome will face another side. Okay, so we always say the keyword or standard phrase that we describe is independent assortment of homologous chromosome during this metaphase 1. How does this process increase GV?
What's this new
Combinations of what? What's the key phrase?
maternal and paternal gametes?
Maternal and paternal chromosomes. Okay, so you form new combinations of maternal and paternal chromosomes. What is another way that independent assortment also helps to increase GV? You can form how many possible combinations of gametes? 2 to the power of B.
11.
Huh? 11.
Yeah.
N. And to the power of N, right? Agree? For humans will be what? Humans will be what? How many possible gametes? How many chromosomes you have? You don't know your own species chromosome number.
With 31.
What kind of species of Homo sapien are you? You have 46.
46.
You have 46 chromosomes. So N represents haploid, right? So
Okay.
how many possible gametes you can form?
22 to about
23 possible gametes. Understand? So remember, independent assortment, how do they increase GV? Number 1, you can form 2 to the power of N possible number of gametes. And number 2, you say you form new combinations of maternal and paternal chromosomes. Next one, the third way that actually increase GV, contribute to GV is what, Ashley?
Random fertilization.
Your head. That is not part of meiosis. So it's what? I joined 2 cells already, that means it's the second meiosis phase already, yeah? So it's what? It's what? Sometimes you're this one, you undergo your this crossing over, right? And then the sister chromatids, are they going to be identical? Are they going to be identical for your sister chromatids? No. So this under metaphase 1. What am I trying to showcase for metaphase 1? Random? Random what? Random orientation. Okay. So the next one is random arrangement or independent orientation of non-identical sister chromatids. Okay. Along the metaphase plate during metaphase 2. So I repeat, random orientation of non-identical sister chromatids. Do you see how each one, they face at different poles? It really matters here. Of non-identical sister chromatids along the metaphase plate during your this metaphase 2. Okay, so this also can form new combinations of gametes. Alright, so learn this out. So this is under your cell division. Okay, now, so how come we learn this? Because we learn your this cell division, you can actually be tested on your this chromosome mutation or gene mutation, right? Agree? So again, I'm only going to go through the main things. Ready? Before we go through a bit of recap, because there's so many topics that we're going to go through today. Or should I do a recap now? Okay, Michelle. First one, can you tell me, Hannah? Usually, only in animal cell, you have this particular organelle involved in cell division called what? Centrioles. How do you describe centrioles? Satu, dua. First one is what? A pair? To each other. Second one is what, dear? Made up of?
9 triplets of microtubules arranged in a
Very good. Next one. Your centromeres is actually going to be complementary in shape to two proteins. Name me one protein. Cohesive protein. But the second protein is called? Please take note. Number one factor we talk about your centromeres, right, is always kinetochore. Try to refrain to write cohesive proteins unless this is actually 3 marks or more. Understand? What is the typical way that we write for exam for the function of your centromeres?
It allows for the attachment of kinetochore proteins, which allow for attachment of spindle fibers.
To itself. For one reason, and you tell me the second reason. First reason is what?
For
Proper
alignment of chromosomes at the equator.
Keyword is proper alignment of chromosomes along the equator, and that will help you in what checkpoint?
M checkpoint.
Very good. Second one is what? Yes? Ensure
No, equal separation.
of
Sister chromatids.
or chromosomes to opposite poles of the cell. Can you now tell me what else does it also associate to? For which other protein? Just now we talked about kinetochore, so centromeres also associate with cohesive protein. What's the number one reason? You cannot just write associate cohesive protein. For sister
Chromatids
to attach. Why? For what purpose? So they can align.
on the metaphase plate.
Okay, in a. Oi, what's the phrase that I've been saying? Proper.
Proper alignment.
Along the.
Metaphase.
Okay, next one. Can you tell me? Yes, Hannah? You ready? Can multitask, right? Okay, we have all the phases, right? Can you tell me? G1, G2, and your M checkpoint. There are 2 overall main purpose of having all these checkpoints. First checkpoint is what, dear?
Give sufficient time for the
Cell machinery to repair. Should there be any? Next one. PPP.
Prevent premature progression
Of the?
cell cycle.
Very good. Next one. We also know that your this meiosis can actually increase your GV, right? How do you usually describe for why do we increase GV? What's the purpose of increasing GV? To increase changes.
Chances of survival,
Continue.
especially during changes in environmental conditions.
Especially during changes in environmental conditions.
So can be selected for natural selection.
For natural selection to operate on. First one, what name me one that contributes to GV?
Crossing over of non-sister chromatids between homologous chromosomes in prophase 1.
How does that actually increase GV? Multitask. Crossing over. So is what?
New combination of value.
Very good. Next one. What is the second one that contributes to GV?
Independent of chromosomes.
Oh,
Homologous chromosomes.
during.
Yeah, the phase 1.
Next. Why? How does that increase GV?
New combinations
Of. And? Next one. What else? Also increase. Form new. How many combinations of gametes? To the power of n combinations of gametes. What's the third one that contributes to GV?
random
Orientation.
orientation
Of?
chromosomes.
Sorry? Random orientation or what?
Chromosomes.
No. Keyword is what? What's the keyword?
Non-sister chromosomes.
No.
Non-identical sister.
Non-identical sister chromatids along the metaphase plate. Understand? Now, so let's say later I will do a summary. I want to talk about these 2 types of mutation and discuss a bit further. So for your this mutation, I want you to take note. Usually mutation, if it does occur, is usually during your this S phase. That's why you can see how it's related to this cell division here. However, I want to ask for your gene mutation. Let's learn certain causes of gene mutation because we know the gene mutation I have is usually substitution, deletion, and this insertion mutation. Okay, can you now tell me for your this causes of your this gene mutation? I said there are a few causes for you to take note of, right? What's the first cause? Can you tell me, Jude? Of gene mutation. What can cause gene mutation? Like for example, what can cause a substitution mutation? Errors due to errors in complementary base pairing. So the first one is that we can have errors in during complementary base pairing where incorrect nucleotide is inserted. This will lead to substitution mutation, right? What else can you tell me that can lead to deletion mutation? Your DNA polymerase skips a nucleotide. What else do we also have for insertion is when this DNA polymerase actually incorporate an additional nucleotide. However, there is one property DNA polymerase always have, which is your proofreading. So that's why another one that can cause this mutation is this errors in proofreading ability. Question, how come DNA polymerase, what are the things that DNA polymerase have to allow for proofreading during in DNA replication? They have a 3' to 5' to 5' X. So as well as a 5' to 3' exonuclease. They are different now. I say one is 3' to 5', one is 5' to 3'. They have just different mechanism, but they all can cleave DNA. Okay, so all this is for point mutation. Okay, so this is going to be the cause, right? So my question is, you know your substitution, deletion, insertion, mutation. They tend to have the consequence of 3 types of overall mutation that you have in your notes, which is here, right? Remember, this is not the type of gene mutation. This is the effects when you have substitution, deletion, insertion, mutation. So my question is, which kind of mutation is more likely going to lead to this missense mutation and why? Can you tell me why? Which one? Deletion only? And insertion, because they tend to undergo and form what kind of mutation as a consequence? We call that mutation. Okay, let's ask. So the question is, okay, just now I asked her, right? How come deletion mutation is more likely to produce a non-functional protein, like a missense mutation? Why? What is the phrase, what we say and discuss about deletion and insertion mutation? When you have deletion and insertion mutation, it result in what kind of mutation? Frame. So how do you describe frame shift mutation here? By the way, it's on this page of your notes, second page, first column. If you want to write the phrase that you every time discuss about frame shift mutation and why you can actually lead to a non-functional protein, okay, that means multiple missense mutation. What is it, the way we write? Because when you have a frame shift mutation, what happens? Any idea? Not sure? You? Cheryl? What happens to all the nucleotides downstream when you have the frame shift mutation? No shit, I also know change, but what is the way that you describe that the nucleotides downstream will be keyword? Improperly grouped. Never mind, write down. So again, can you please take note? The way you describe frame shift mutation, why there's a higher chance in forming non-functional protein or many missense mutation is, number 1, you say nucleotides downstream, keyword, are improperly grouped into codons. I repeat, this is the first key phrase you tend to write for exam. I repeat, nucleotides downstream will be improperly grouped into codons. Okay, yes, you want me to say that? You have already? Okay, then second thing is what? Ashley? Is what? Your lead 2? What kind of mutation? No shit, I want to know what kind of missense. Extensive. Leading to or resulting in extensive, okay, mutation, which change the 3D conformation of protein. Keyword, underline, extensive missense mutation. Okay? Question. Inversion also can, the idea you should know, but usually these are the 3 major ones they like to test. But inversion can, okay? So later we'll discuss how they also can lead to like cancer or mutation in your proto-oncogenes or your tumor suppressor genes. Anyway, can you tell me, Yunshi, how come your base pair substitution is less likely to produce a non-functional protein? So what kind of mutation it result in? Silent mutation. So when you have silent mutation, what is the phrase? How come can lead to silent mutation? So you always must discuss, because the genetic code is whether same amino
acid can code for the same codon.
or by the same code, by a same codon, by more than one codon.
Yeah, mine.
So due to what effect?
Local phase.
wobble-based effect. Understand? Next one. Can you tell me, Ethan, how come substitution mutation, okay, can also not produce a non-functional or less likely to produce a non-functional protein besides silent mutation? What if the mutation is missense?
Then it can result in a change in amino acid with similar R group properties.
Yes, so sometimes when you have substitution mutation, you can actually replace the amino acid that has similar R group properties. So what does it mean about my this 3D conformation? Unchanged. So the 3D conformation most likely is unchanged, so the function most likely is also unchanged. What else, Jude? For substitution, how come less likely to produce a non-functional protein? Even though missense? Because, let's say I told you, maybe you substitute at a region that is the non-critical region. Do you know what your friend just said? And then the idea is like I told you, right? Let's say we have enzyme. The critical region is always the active site. Maybe they do outside of the active site. So that's why, right, isn't that not going to change the active site shape? So again, you can say missense mutation occurs in the non-critical region of your this protein, or you can say it's a non-essential amino acid. So that's why the function of the protein should not be affected. Understand? Next one, I want to talk about two types of chromosomal mutation, their causes. You know the two types of chromosomal mutation are, number one, we call it aneuploidy, which is always due to, the phrase that is always related to aneuploidy is what, Ashley? Hello, can you please? Okay, this is always the phrase side by side. Now let's ask, what's the causes of aneuploidy? So Ashley asks, what do you think can be a cause that can lead to non-disjunction or lead to aneuploidy? Because we know non-disjunction means failure to separate, right? So what could be a possible cause there?
Unequal separation.
Unequal separation. I know what is unequal separation, but I'm asking what can lead. So what do you think I'm testing you on? I want you to make sure you can think about beyond other chapters. It doesn't mean they strictly will test you on one chapter. What do you think this is also related to? Cell?
Division.
Cell division. Okay, so what do you think can happen during your this cell division? Some mutation or errors must have occurred during cell division.
Mutation of the kinetochore.
Okay, so what happens to the kinetochore?
Cannot attach to the spindle fiber.
So again, so many things, right? So for example, you can say gene mutation to what? To kinetochore proteins or cohesive proteins. This can be one, right? Agree? I also can have your this substitution mutation or your this whatever mutation to your this centromere. I cannot call that a gene because centromere, you should know, is not coding for any protein. But the idea is that how come mutation in centromere can lead to aneuploidy? Maybe your mutation in centromere will change the conformation shape of your this centromere, doesn't allow or disallows your this kinetochore and your cohesive protein, okay, to bind. Okay, so next one, what else? So for example, what can we also have? Maybe microtubules usually must attach to kinetochore, right? What if we have improper attachment? So I'm giving you all some to ponder because I want you to make sure that sometimes you can think beyond. Okay, improper attachment of microtubules to kinetochore. Okay, what else can we also say? Remember there is also another proteins that is involved in cell division. What is it, Ashley? What's another cell proteins also involved in cell division? Cohesin, right? So usually during anaphase, what must cohesin undergo? Cleavage. So what if your cohesin proteins did not cleave? So again, you can say cohesin protein complex not degraded or cleaved. What else can I also say? Your centrioles. Maybe we can have, again, your this multiple centrioles or mutation in centrioles or absence of centrioles. If you have multiple centrioles, you should know that I can have, again, multiple mitoic spindle apparatus. Wouldn't that also lead to aneuploidy? If you have absent centrioles, that means you also cannot synthesize microtubules. What? You also lead to aneuploidy. So you can think of multiple ways. There are many ways because all this is supposed to help in your suggest question. That is in your this paper 3. Understand? Structural mutation, you should know already. Usually, this one is anaphase, right? So structural mutation is usually in what phase? Can you tell me, Hannah? Before I go through the differences and similarities between gene mutation and chromosomal mutation. When can this actually occur in cell division? Errors in prophase 1. Understand? For example, what can actually be a cause? Think about this. In prophase 1, what do we typically undergo? Hannah? Or between? Off. So what if you have unequal crossing over due to maybe misalignment? Misalignment, okay, of my this homologous chromosome, such that you can lead to, let's say, your insertion, translocation, mutations. What else can also be a cause for your this, again, structural mutation, Cheryl? Usually what kind of chromosomes can pair up together? But what if, again, you want to lead to this? Maybe you have crossing over between non. How come we can lead to structural mutation? I'm thinking about how can we lead to here. Maybe crossing over between non-homologous chromosomes. By right, they shouldn't even undergo crossing over. But I'm just saying, you can actually think outside of the box. All these are the various ways, okay, for you to try to expand your idea, right, to see that how, okay, or what could cause all this kind of different kinds of mutation. Understand? Now, I want to talk about your similarities and differences of your gene mutation as well as your chromosomal mutation. Ready? Can I erase? Eraser? I'll do this as a table because by now you should know already. I'd like to rationalize your learning. Tables are the easiest way. Eraser? Okay. Let's talk about the similarities. There are many similarities between your gene mutation and chromosomal mutation. Let's try. First similarity. First similarity, what do we learn? We learn that both can actually change and alter your this genotype, alright? Or we can say DNA sequence. So you can use this for, again, your revision. So both may involve. Notice how I write may because you know gene mutation sometimes can lead to silent mutation. So changes in your DNA sequences. So find some space. So you can think there's a lot of space at your gene mutation at the back. You can actually write that. Or your this genotype as well. Alright? So please take note, I'm talking about DNA nucleotide sequencer. Second one, what's the second similarity I can say between both of them? So both, as you can see, may result in, again, disease, cancer, or maybe abnormalities. Please refrain from writing saying negative effects. It's very vague, okay? So specify. What other things that we can say between your this gene mutation and chromosomal mutation? So both, do you agree? Potentially, they can change your this proteins produced and therefore your phenotype. Because if you can potentially change genotype, you also can potentially change your phenotype. By the way, this is also how we can arise to multiple alleles. Okay, like IO alleles over time. Okay, so both may result in changes in protein structure, in protein production. Okay? Or you can say phenotype, I can also accept. Another one. Both may also give rise to new alleles or variation in the population. Or you at least tell me variation in the population. But the question is, let's ask, can you tell me, Cheryl, which one is more significant impact in evolution and why? Between gene mutation and chromosomal mutation, which one has more significant impact and why?
Gene.
Gene mutation, why? Because you create
New
alleles. But this one is just yes, that means reshuffling alleles already existing in the gene pool. But how come I say this one also can give rise to new alleles? Like you think about your this structure mutation or your this again, whatever mutation. How come chromosomal mutation, by the way, still can give rise to new alleles? Any idea? Yunshin? What about the gene loci? But that also is reshuffling repositioning the allele to another location, right? If that's what you are talking about. You know what I'm talking about? Which
If
kind?
you exalt in the middle of the gene loci,
That means it's what kind of mutation? Which one do we look out for?
translocation.
Cannot be
Unequal
translocation.
crossing over.
No, I'm talking about what kind of chromosomal mutation can give rise to not new combination, but new alleles. Inversion mutation.
Oh.
Structural inversion mutation, when you sweep, the whole thing will be read in a reverse manner. Potentially, it can create new proteins. What? Isn't that going to be, can potentially give rise to new alleles? So it doesn't mean it only can give you the new combination. So you can think about that. Clear? Next one. Both, hopefully you know, okay? They affect germline and somatic cell. Both are affected. Germ cells are actually your sex cells, okay? And somatic cells. We also know that this one can be also inherited in offspring. Both mutation can be inherited in the offspring. Okay? Also you should know both are actually just spontaneous random processes. Okay? Alright? Those we also can take note is that both may also be caused by mutagens. Or you can say radiation. Well, there are differences, right? So let's talk about the differences between your gene mutation and your this chromosomal mutation. You should know already, gene mutation, right? We are purposely just changing the nucleotide sequence on your DNA. But this usually change your this structure or your DNA, okay, or your number of DNA. So this one involves changes in nucleotide sequence of DNA. You should know this one can change again in structure. Okay, or numbers of chromosomes. You should know already, for your this gene mutation tends to only affect one gene locus or one gene. But this one affects more than one gene locus. Multiple gene loci at the same time. So, please take note. Oh, I forgot to write. Nucleotide sequence mostly unchanged. Okay. Another difference I also can talk about is again caused by what? So hopefully you know this is actually, let me write another color. Sorry, I forgot to write the other color just now. So this one is caused by, okay, random error during DNA replication. But potentially, we also can be caused by damage, okay, due to presence of mutagens or radiation, okay. DNA due to radiation or presence of mutagens. For this one, you should know already, caused by errors in crossing over, okay, or non-disjunction. Okay, of chromosomes when they fail to separate. Or you can say how, again, you can segment of chromosomes break and rejoin onto another chromosomes, alright, during or crossing over. So, or again, unequal crossing over. Or errors in crossing over. Okay, another difference is that we can talk about the effect. Okay, what kind of effect it can result in. Gene mutation can result in your this silent, missense, or nonsense mutation. Let's do a recap. Whenever we want to discuss nonsense mutation, there are always key phrases that is related to the word nonsense mutation. Nonsense mutation, you will always now code for a premature stop codon, which is your UAA, UGA, or UAG. And then what's the keyword that you always describe about the protein? Protein is always going to be, can we just write shorten? What's the T word?
I forgot.
Run.
Or truncated
Protein. Can I just write truncated protein? I emphasized this last week.
No. Non-functional.
Yes, you must write truncated and non-functional protein. Okay? Whereas this one, you should know already. This one can actually, oh sorry, I forgot to say. For this one, right, you can have either silent mutation can result in no phenotype changes as well. Okay? Phenotype may not change. Okay? We already understand why, even your missense mutation also showcase why. Okay? But this one, phenotype mutation, usually can change. Yes, so usually will cause disease and change phenotype. Or I'll say change phenotype. How come this one is more likely to change your phenotype? You know? How come chromosomal mutation is more likely to change phenotype? Because it affects multiple gene loci and therefore multiple genes. So that's why the extensiveness of mutation is much more greater. Understand? Next one. This one you can see it can result in new alleles, alright? But this one reshuffle alleles already existing in the gene pool. So in other words, the mutation, right, when they have change in phenotypes, how come change in phenotypes? Let me explain. Because even though your nucleotide sequence mostly unchanged, we can change the level of expression of your this genes. Sometimes there may be overproduction, underproduction, or even no production at all. Clear? Last one I'll just write, not very solid, but still is still a difference. This one definitely cannot be observed in your karyotype. But this one can be observed in your this karyotype here. Who got questions now? Again, I'm only going to go through the main things. So know the difference between gene mutation and chromosomal mutation. Now, why do we study mutation? It's going to be related to proto-oncogenes genes, tumor suppressor genes, as well as your this telomerase gene. Sorry?
What's the
Karyotype is like the whole your number of chromosomes, okay, in a single individual. So they will showcase all the 46 chromosomes here. So imagine if you're aneuploidy like Down syndrome, you have extra, right? Agree? By the way, it's in your syllabus to know an example of all these different types of mutation. What is a disease that can showcase for gene mutation and know how to explain? Because in essay, sometimes you must say, right, with a name disease and describe, right? What is one disease? Sickle cell anemia, where you have what kind of mutation? Substitution mutation of a CTT become CAT. CAT, right? Agree? So CTT is substituted as CAT. So what will happen to my this what do you call that? Why am I tripping? What do I want to say? Oh, what's the change in amino acid? Yes. What's the missense mutation? What's the change in amino acid? Sorry? Glutamic acid, which is actually a charged R group, is now being replaced and substituted with valine, which has a what kind of R group? So what happens to solubility of your sickle cell? For your hemoglobin? Less soluble. By the way, let's do a recap of hemoglobin. Hemoglobin actually have high. Let me repeat. Hemoglobin actually has two structure features that enables its transport of oxygen. What's the first structure feature that I emphasize? How many heme groups?
4.
Which one? 4, 6, 8, 10. Is it? No. Hello, it's 4. So 4 heme groups enable each hemoglobin molecule to bind to how many oxygen? Hello. 1, 2, 3, 4. So it's what? So 4 heme groups enable the binding of how many oxygen? 4 oxygen molecules. So what is the property it confers? High oxygen carrying capacity. Again, your hemoglobin has high
Oxygen carrying capacity.
due to presence of.
4 heme groups.
Okay, what's the second property of your this hemoglobin that we learned? Don't know? What's the second property of hemoglobin?
Cooperative binding.
Yes. How do you describe an exam? Binding of?
There's like no disulfide bridges between each of the 4 subunits of
So?
hemoglobin, so then they can move with respect to one another. And
No.
binding of
What do you
one-
write in exam? If let's say this is just 1 mark on cooperative binding, how do you
binding
describe?
Of, one oxygen to one subunit increases the affinity of the other subunits for oxygen.
Due to change in?
Conformation, 3D conformation.
So that's why, right, when you actually bind, you change in 3D conformation of the other subunit, increasing the loading of oxygen. This is called cooperative binding. Can you tell me, just a quick one, this is for your MCQ only, every, or even your this essay, every hemoglobin is an alpha beta dimer, right? It's a quaternary structure, right? So your alpha is made up of how many amino acids?
What? Okay, that can't be a real question.
Oh yeah, it's a real question, I can show you. So it's what? 100 and? It's for essay. 41. 141 and 146 for your beta.
No way.
So please take note, this is for your primary structure. So need to learn, because I've seen in an essay question before, they ask you to describe the protein structure of hemoglobin. Then primary structure you must take, and then that will give you a 1 mark. Okay, now next one, can I ask? For this, why do we study gene mutation and chromosomal mutation? Because we need to learn your this tumor formation and your this cancer formation. I'm not going to go through what is the difference between cancer cells and your normal cells. I expect everybody to know what is the difference already. But what I want to talk about is the most common question they like to ask is, again, describe what are the key features for the development of either sometimes they use the word tumor, sometimes they use cancer formation. Because is tumor cancer, by the way, Ashley?
No.
No, not necessarily yet. But one thing is, both of them, if you want to become cancer, there are some key features that we describe for cancer. If this is actually cancer, what do we learn? Some key characteristics of cancer cells there. So your these cancer cells, you can have uncontrolled cell division. Continue. Because that is also for tumor.
Angiogenesis.
I want to see things called metastasis. If you want to describe your this cancer, that means where you actually break off and actually cause secondary tumors at distant location. Alright, what else? Angiogenesis is also other key phrase that you always see in your answer sheet. If you ever see a question that inside the question itself, describe how it caused the development of cancer. Understand? Now, again, when do you use uncontrollable cell division? All the time. Because both tumor and cancer formation, you have uncontrollable cell division. And then the other key phrase that is always related with the uncontrollable cell division is what? Again, you have loose, okay, contact inhibition. This is when it's talking about tumor, talking about cancer, always write this here, okay? But if it's cancer alone, this is always the characteristics of cancer cells. However, what are some other two more key features that we always describe that is related to the word tumor formation or cancer formation? Hannah, I mentioned this before. I don't know whether you recall.
Accumulation of mutations in a single cell.
Very good. So again, I always want to see accumulation of mutation within a single cell lineage. All right. Keyword and emphasis you will see in exam is called a single cell lineage. Okay. So I repeat. I always want to see accumulation of mutation within a single cell lineage. It's not a single cell, it's within the same lineage itself. So I always want to see how you have many mutations. For just tumor alone, we always need to have gain of function of how many proto-oncogenes at least. Cheryl?
2 oncogene.
And how many loss of function mutation of tumor suppressor genes? Get ready, I want you to tell me what is the difference. 1, 2, 3, the three differences between proto-oncogenes and the tumor suppressor genes. Of how many tumor suppressor?
1. No.
You always need at least one proto-oncogene to mutate to become an oncogene. Okay? And then you need actually loss of function, oh sorry, I forgot to write mutation, of many tumor suppressor genes. These are always the keywords you will see appear in exam whenever they ask you to describe how does this lead to the development of cancer formation or tumor formation. Understand? Ready? But the question is, are you sure that you just need 1 or 2 tumor suppressor gene, like just need one P53 and another tumor suppressor gene to become cancer? Or are you talking about 2 alleles? Which one? Because 2 alleles to knock out that gene is the one I think you are referring to as 2. Do you know what alleles? Yes, but you are talking about many genes. That means I need a P53, I need, let's say, an SMAT2, I need so many other tumor suppressor genes to actually get knocked out. And then that can possibly lead to the development of cancer because you will miss all the cell checkpoints, which are G1, G2, and your M checkpoints. Understand?
Actually, many types, not be just 53.
Sorry again?
Must be like a lot of types. Or can it be the
Or
same
tumor
type?
suppressor genes?
Yeah.
Yes. A lot of different tumor suppressor genes that undergo mutation. That's the idea. Okay? Okay, come. Dude, can you tell me what is the you. What's the first difference between proto-oncogenes and the tumor suppressor genes? I went through under your this table before. Can you name me one? Between tumor suppressor genes and proto-oncogenes?
genes encode for genes that
Encode for proteins, you don't encode for genes. Encode for proteins that?
stimulate cell growth.
Wrong, I will not accept cell growth. Cell growth is not the same as cell division. Why? The name itself. Cell growth.
Multiplication. I don't know.
Cell growth.
Uh-huh.
So the cell become?
Bigger.
Yes, cell division means you divide to form multiple copies. So I will not accept cell growth, you understand? So they stimulate, sorry, not stimulate, generally encode for proteins that stimulate what? Cell division. We have many different kinds of proto-oncogenes, right? For example, what could be a proto-oncogene? RAS protein that is involved, because RAS is a type of G protein, right? Agree? Isn't that part of cell signaling that brings about the cell division? Can I also say the receptor, like a G protein coupled receptor, is it also a proto-oncogene?
Yes.
Yes, any proteins that are part of normal cell division, they are actually proto-oncogene. You understand? Yes. So your protein kinase, it also can be a proto-oncogene. Clear? Clear? Next, how about tumor suppressor gene?
It encodes for
Proteins that?
inhibit cell division.
How? By arresting
Oh,
cell
cell
cycle, promote DNA repair, and they also stimulate apoptosis. Now, in your notes, do you see your this cancer? Last page of your notes. Please at least know the 3 roles of your this tumor suppressor genes. So I repeat, they can arrest the cell cycle, they can actually promote apoptosis and also promote the synthesis of enzymes that will bring about DNA repair. So what are some of your this tumor suppressor genes we learn? TSG. But there are many different kinds of tumor suppressor genes, okay? As long as they are, like say, your cell cycle check regulators, isn't that also a tumor suppressor gene? Anything that promote or inhibit your this cell division, okay? Okay, if it promote apoptosis or inhibit cell division, they are part of your tumor suppressor genes. Understand? Give me another difference between proto-oncogenes and tumor suppressor genes.
There's a gain in function mutation and a loss in function mutation.
So how many do you need to actually have mutation of?
One copy of the gene needs to be mutated for proto-oncogene, but for tumor suppressor gene, both copies need to be mutated.
So again, another way he can also write is that for your tumor suppressor genes, it acts recessively. Mutation acts recessively. That means both of the alleles need to be actually knocked out in order to promote your this tumor development. Whereas we also can say mutation acts dominantly for your proto-oncogenes because only one of the 2 alleles need to be knocked out in order to induce cancer. What's another difference? Results from? Because I want to bring about to that. Results from?
One of them is from chromosomal.
Are you sure one of them must be always chromosomal?
No, like sometimes.
Sometimes. So gene mutation cannot bring about mutation in your this. Can ah? Then how Suggest? Huh? What?
So hard.
So hard? Not hard. Why you hard? No hard. Yeah, let me write here. Type of mutation first. So we can actually take note how we can actually bring about cancer. Some of you, like you, I expect you to know.
Teacher, do you have lightning charger?
Lightning charger. Lightning.
Oh, thank you.
Okay, now, anyway, I write POC first. So you should know already, POC is proto-oncogenes. Please know how to describe. Okay, so let's talk about what kind of mutation can bring about your this mutation of your proto-oncogenes. So you can not only use this to study for your essay, but you also can use this to study for your paper 2 or paper 3. Okay, so let's try. So the type of mutation we have is again your this chromosomal mutation. Let's talk about your structural chromosomal mutation. So one of the mutation we have is your translocation mutation. Okay, so this is structural chromosomal mutation. So what is your this translocation mutation? You hopefully know translocation mutation is when a section, I'll just write here, So again, it's when a section, okay, of your this chromosome breaks off. and attaches to a non-homologous chromosome. However, how is it going to be related to the gain of function mutation of a proto-oncogene? So one reason is that for this proto-oncogene, so the POC, alright, may be moved to where? From its original location in one chromosome to another, where it's now under the influence of enhancers or a more active promoter. Get ready, yeah? 1, 2, 3. Tell me how enhancers increase the rate or upregulate transcription. Okay? Alright. Okay, ready? How does your this enhancer will be what do you call that? Increase or upregulate your this transcription?
Recruits general transcription.
Alright, DNA bending proteins. 1, 2. Wait, your syllabus. I went through this last week. 2. Bend the DNA. Why must we bend the DNA? Can you tell me? Standard answer. To increase interaction between what? Activators and other transcription factor. Again with more fluency and charisma. So it's what? Recruit
DNA bending protein
to bend
to bend the DNA
in order to increase
increase interaction
between
between activity
and
transcription factor.
other transcription factors. Next, Cheryl, haven't done yet. This will increase the of the transcription initiation. And then what do you end up with? This will What are you talking? Increase affinity. Upregulate transcription. That's all. Understand? Okay, next. What other mutation can I also bring about? Can you tell me, Nathan? That can bring about POC?
Substitution.
I want still structural mutation.
Or
Suggest?
insertional mutation.
Insert now.
Yeah.
Can. Insertion mutation. I will write duplication or insertion. They are both under the same thing. Okay? So idea is, hopefully you know what is duplication now. So duplication, I'll just write the meaning first. Okay? So where it involves replication of a section of chromosome. When you replicate this section of chromosome, resulting in increased copies of the idea that certain genes.
Your phone is at 90%.
Oh no, give me a moment. Thank you. Genes, so you can say again a set of gene loci being repeated. So how come does this lead to actually your POC becoming your this proto-oncogenes? So this time it means that you have multiple copies of the POC. Okay, so multiple copies of the proto-oncogene, you will have increased expression And when you increase expression of the gene, you have excess of this protein that stimulates cell division. Idea is multiple copies, either you increase expression and overproduction.
Why don't you repeat a set of gene loci?
And because duplicate one.
So it got
Chromosome
two.
is a gene loci, no? Do you see? That means this one, you actually, some mutation happen. You have one set, you have another set. Isn't that a set of gene loci? Okay? Next one. What else can we also have besides translocation mutation? Can you tell me, Jude?
Yeah, inversion.
So we can also have inversion mutation. Inversion is a structural mutation. Can you describe for me how come we can have inversion? What is inversion there?
It's when two set of gene loci.
So the idea is the set of nucleotide sequence of this gene loci is actually inverted, right? Okay, so again a set of gene loci inverted. Wait. Why would this inversion will actually bring about top to become oncogene?
Oh,
Makes no sense. That is for tumor suppressor gene, right? So, is there any more?
God.
Forgot? Ashley, tell him. Is there any more? Sorry? Aneuploidy. So we also can have aneuploidy. Same thing, okay? Non-disjunction. Okay, led to multiple copies of proto-oncogene. In a cell. So that's why we will say how this increased expression of genes, because now you have multiple copies and therefore overproduction. Okay, of proteins that stimulate cell division. Actually, can gene mutation also bring about proto-oncogene to become oncogene? Can. So what kind of mutation? So now so far we did chromosomal mutation. I'm going to do this gene mutation now. So why? Gene mutation and where? That can become your proto-oncogene become oncogene. Enhancer. So again, gene mutation can occur in two ways. Number one, mutation in the enhancer or silencer. So when I have mutation in the enhancer or the silencer or the promoter, how come we can lead to formation of oncogene? Ashley?
Increase expression of your proto-oncogene.
So the idea is that you want to increase expression. Okay, what other mutations, gene mutation I can have to become a proto-oncogene? Hannah? Is that? You also can have mutation in where? Can I also have mutation in my exons to become a proto-oncogene to an oncogene? Can, right? There's a variety of ways, right? So mutation occurs in exons. Okay? Or you're this, again, you're the nucleotide sequencer. Alright, so whenever you change your nucleotide, it will actually alter the 3D conformation of the protein coded for, such that it's now hyperactive. I'm going to give you an example. Either you say hyperactive or is it resistant to degradation. Example, you have mutation in your receptor. Okay, so mutation in a receptor here. Oh, can you give me a moment? My Queen Dowager is here. Give me a moment. She said Queen Dowager. Queen Dowager. Wait, what was Queen Dowager? Very stupid. Yeah,
She doesn't really more than you.
I don't know. Maybe I have tuition.
Ooh. We should do that. Almost like it's not her day, like what you did to me last time. So pains, yeah.
Okay. So for example, I can have, let's say, your this cell signaling, your this the cell that undergoes your this receptor, like your G protein coupled receptor. An example, maybe I can have mutation in the G protein coupled receptor such that this time that mutation will cause the receptor to be active even without the binding of a ligand. Wouldn't that also become an oncogene? Or maybe this time your RAS, related RAS, let's say your this RAS can no longer be degraded. Okay, so you cannot shut down the signaling as well. Okay, so these are all examples of proto-oncology. Clear? Next one, your this tumor suppressor gene. Let's learn some mutations that can actually occur. So tumor suppressor genes are actually those that encode for proteins that either promote apoptosis, or again they actually can also promote, let's say, this DNA repair, or even let's say the arrest of your this cell cycle, right? So now again, can I actually have your tumor suppressor gene can undergo gene mutation, since I already add gene mutation, Cheryl? Can gene mutation can bring about your this tumor suppressor genes that become your this knockoff? Can, right? So for example, gene mutation, let's say if I have this, instead of saying increase, can I now say that the gene mutation in, let's say, your this promoter or enhancer, this time will decrease expression? Or you can say how it will actually inhibit the entire expression. Alright, so in decrease expression of my this tumor suppressor gene. This is one. What other mutations I also can have? Mutations I also have with the exons, but this time you alter this. Instead of saying it's becoming hyperactive, you will now say, instead of becoming resistant to degradation, you will say now it's now non-functional. Okay, so this is the difference. Alright, now, but we also can have other mutation, right? So let's try. Duplication definitely is not going to help in your this knocking off of my tumor suppressor gene function. So let's see what other mutation. So one mutation I can bring about is, you should know, translocation mutation. I'm not going to explain what translocation is. I'm just going to simply just state what is the effect on your tumor suppressor genes. So for translocation mutation, so this time we may most likely the tumor suppressor genes is placed where, Yunshin? Most likely can be moved to where? To actually knocked off. So they move to another chromosome that is placed under what? So you can knock off the tumor suppressor gene. Think about your this eukaryotic genome. Okay. You want to place under the control of silencers. So if you place under the control of silencers, or again, like less active promoter, this will actually have lack of expression of the protein and inhibit your cell division. So you can say causing lack of expression from gene product. Okay, so that's why you will have again inhibiting your this cell division. So case under control of silencers or less active promoter, so lack of expression to inhibit cell division. Now we also can have, don't write antibody first, huh? We will have what else? Inversion. So this time inversion is relevant. So for inversion, your structural mutation, do take note, what does it occur? So I'll just write down the reason first. Occurs when a segment of your this chromosome is actually going to be inverted or separated and then placed in a reverse manner. So it's separated and reverse its sequences. Okay, so reverse its sequences and rejoins. Okay, at the original position. So when we have this, one thing please take note, why does this actually can knock out this? So you can see how it can result in formation on non-functional protein. Because do you see the missense mutation will be very extensive, alright? So that's why it's no longer able to inhibit your this cell division or DNA repair. Or again, you can even say how promote apoptosis. Okay, next one. So it's separated, reverse sequence, and then rejoins. This one will lead to non-functional protein. What else? What other mutation? That's why you can see they are all related. Other mutation that can bring about your loss of function of tumor suppressor genes is this deletion mutation. So this one I'm only explain the reason why, so you all should know. So for your this deletion mutation, idea is just telling me you have loss of your this tumor suppressor genes in the deleted segment. So therefore, do you see how I can no longer have expression? So this will just say lack of expression. Okay, so I'll result in lack of expression of my tumor suppressor genes, so that's why I can no longer inhibit. Last one, what else can I also have mutation of? I'll just write translocation mutation. So you should know translocation also can result in this. Not last one, sorry. Second last one, translocation mutation. So all you need to tell me is that you translocate to a...
Usually there.
Oh, I say already. Oh, then aneuploidy. Aneuploidy. Aneuploidy, so same thing, non-disjunction leads to loss of tumor suppressor genes. So non-disjunction leads to loss of tumor suppressor genes. So same thing, oops, sorry. So you just need to take note, this results in lack of expression. Okay, of tumor suppressor genes. Okay, Now, one thing, right, we already learned, so you can see how mutation is going to be related now. So know how is the relation like. But can I do a quick recap? One thing, there's another cell that has in common with that of your tumor cell and your cancer cell, which is your stem cell. But can I say tumor cell and stem cell are the same? So let's ask what's the common or similarities between your this stem cell and tumor cell. 1, 2, 3, 4, 5. First one.
The telomerase activity.
Oh, no, both of them have high telomerase activity. Just a quick one. How do you usually describe the telomerase activity? Elongates. Low. Yeah, so what's the purpose? Why? So you will not shorten to the minimum critical length. So you will actually undergo unlimited rounds of. Okay, next one. Both of them undergo binding. Yes, next one. Stem cells and cancer cells. What do they have in common? So both have high telomerase activity. Both actually have undergone mitosis. What else? Think about that. What else?
Uncontrolled.
Sorry?
Uncontrolled cell division.
You sure that stem cells is uncontrolled? No, right? So stem cells, can I ask, when they actually divide, is it unlimited number of times? So both can undergo
Cell division.
unlimited.
Unlimited cell division.
Yes. Please know the key phrase. Your problem right now is you forgot the key phrase all the time. You, what's another similarity?
Both not differentiated.
So both are undifferentiated. Very good. Last one. Oh crap. Both are undifferentiated. What else? Can you tell me? You're not in tune? Help. What's another similarity? Sorry, what you say? For prolonged periods of time. Sure, fine. Both are like actually acceptable. Understand? But is stem cells actually considered your this cancer cells, Ethan? Okay, come. There are multiple reasons why stem cells is not considered your this cancer cells.
They got the ability to differentiate upon receiving molecular signals.
Very good. That is for stem cells, but your this cancer cells will always.
Cannot differentiate.
So your cancer cells will always remain again undifferentiated. Next, can you tell me another between your this cancer cells and your this stem cells?
Is it stem cells have the contact inhibition?
Stem cells have no contact inhibition, but your this cancer cells have contact inhibition. Is that what you're saying?
No.
So it's what?
No. It's the other way around.
So stem cells have?
Contact. No contact inhibition. Have contact inhibition.
You know what contact inhibition? Contact inhibition, that means when they grow, they touch, they stop dividing already.
Can
So?
stack. Does it mean can stack?
Something like that. So, in other words, cancer cells have contact inhibition or not contact inhibition?
Wait.
It was a
Wait.
simple question. 没有
Have no.
no contact inhibition, but normal cells have?
Have.
Okay, stem cells also has contact inhibition. Next one, can you tell me another difference between cancer cells and stem cells?
One have uncontrollable
Cell,
division.
while your cancer cells, cancer cells have controllable or uncontrollable?
Uncontrollable.
Uncontrollable cell division, while your normal stem cells,
Have controllable.
why? Due to presence of what? Why your this normal stem cells can have controllable cell division? Due to presence of check
Oh, checkpoint.
checkpoints and functional.
Optimal suppressor.
Yes. Okay. Name me another difference between your stem cells and cancer cells.
Cancer cells can undergo metastasis.
Metastasis. Okay. Wow.
Stem cells cannot.
Okay. Name me another difference, dear. Oh, Cheryl. Oh, my eyes here. So again, what is another difference between cancer cells and stem cells? Cancer cells, they can undergo that formation of blood vessels to itself. Angiogenesis, but normal stem cells cannot. Last one? Which one? Cancer cells can undergo apoptosis, cancer cells cannot. So please know the difference. Okay, come. Can we try some questions? Can we pass them? I want to do the cell division as well as your mutation and cancer. and pass down, please. By the way, each of you will do your respective questions for me to mark. Do you see we have question 2, A, B, oh, do you see we have entire question 2? So again, Hannah, Yunxin, and Jude, I want you to do question 2A, B, and D. Okay? Now the rest of you, do you see we have question 1A as well as question, again, 3B, sorry, 3A and 3B. Can you help me do question 1A and 3A and 3B first? One A and 3A and 3B. Once you're done, let me mark. Always look at the keywords, then you see how the marks are delegated accordingly. So fierce. Okay. Don't cry, dear. Go sweet. Take a sweet. Oh, by the way, where's your paper? Can I pass you your paper? The practice paper?
Me?
Yeah.
What practice paper? This one, is it?
Oh, yes. Okay.
Ah.
So I already given our answers as well, so we're going to just have a look.
Oh no, for so many prompts.
So I already gave my 2 cents why. I will be giving out paper. Give me a moment. I'm going to print out.
20.
Why? We print out. This is another practice paper I want you all to try. Then later, if you want me to mark, feel free to give me next week. Give me a moment. So I'll give you the soft copy, so if you want to complete, give me by next week. The rest of you I'm printing out for you, so if you want me to mark, you just send me by next week. Hard copy, soft copy, either. Everybody get.
Yeah, I think we did it already.
Oh, good. Then let me mark.
No.
Why?
Wait, what's this paper? Is it like
Paper 3.
like is it like you compile the question?
No, it's a printing paper.
Wait,
So
which
again,
two? I think I did already. Like I think I did in school.
you did in school. I can give you another, then you complete for me.
No, don't want.
Why you don't want?
I don't
Then you
know.
can practice your writing because you can see your writing right is actually very it needs work lah.
The essay.
Yep,
Wait.
especially. If you want, then you can complete the essay for me
Is this-
because
is
I'm
this-
sure that you
is
haven't
this
done the
four?
essay. What?
Is it 4?
4 marks, yes.
Is it 9?
You don't be stupid, right?
Whatever your friends say. But I can't say it because I don't want to end on mothership. So anyway, haha. So whatever your friends say. Take one. If you want, then you can go and try this essay. Okay. Isabel, I'll send you the soft copy. Have you done the NYJC paper, dear? Isabel, or you have done it and then you want to try another paper?
Wait, NY like
NYJC 2023, paper 3.
wait, let me check.
Very hardworking, means you've done a lot now.
Well, I actually did this already.
For
Like,
essay?
I marked it already. Yeah, like I did like the whole thing.
Oh, even essay. Okay, give me. I'll give you another
Is
paper.
this like, wait, is this like NY or
Mm.
is it like 2023?
So I give you.
No, the one.
Why you?
Wait,
Huh?
I haven't done the NY.
You haven't done NY, then you can complete for me. I will send you the soft copy right now. But you have, right? Actually, you have. Then you just call. Okay, now I just send you the soft copy. You can do it through the soft copy and just send me when you're done now.
Okay, thank you.
Because some of the NY paper is the answer is quite vague now. Sure, you even did the essay,
Mm.
Then how you
Mummy?
score for this paper?
Not good.
This
Like-
one is you do it in your school practice
No,
or?
it's like I do myself one.
Okay.
But like,
Please understand now. Okay.
yeah, I get like 50 something.
You know what's your aim, right?
Wait, no, this is paper 3, right?
Yes, paper
I think,
three.
I think I did better. Like,
Mm. You did is what?
wait, it's out of 75, right? This one.
Mm.
Oh yeah, I didn't do it, I got like 40.
Every time you 75, right? By right, for your paper 3, you should actually aim for like 58 and above.
Oh, 58 then high.
Then paper 1, you should be aiming, okay? It's like 25 and
Paper 1,
above.
paper 1 is MCQ.
Paper 2, huh?
Paper 1 MCQ.
Yeah,
25.
paper 1 MCQ. And then
25
paper 2
then
by
not
right,
good.
you should be aiming for let's say 60 plus and above for paper 2.
Luckily,
Because
okay.
together with your this paper 4, everything, this is the A if you are aiming for A.
Do you know about paper 4?
Paper 4 is out of how many again now?
Yeah, 55.
55. So 55 usually you should be aiming for the 45 mark.
Wait, but it's damn hard. Bio grad damn hard.
Huh?
No, like, wait, is it not? Like, I feel like it's damn hard, yeah.
For this one your planning is what this time around?
Bro, my planning is like, I mean
Yes,
my planning like, I got like 4 or 12. Then it's like, I studied so hard for planning, you know, then like,
Okay, so I told you already, right?
Wait, like,
The
but then,
idea
like-
is a markdown scheme, what?
I know, but
So
I
have
feel
you
like I did damn
realised?
well. Like,
Okay, then why not you go in retrospect this time? See what are you lacking. If you still don't understand, then you send me. Then you ask me, Archer, what do you think I'm lacking?
yeah, I feel like I don't know, I feel like my school marking scheme is like very strict. Like, for
Have to
prep.
be strict, what? Prelim
No,
is
no, no,
usually
like
extra
not
strict.
straight, like not straight, like damn sort.
Or
like, it's like that.
psychotic, huh?
Yeah, yeah.
But sometimes having a psychotic teacher marking is better than again
No, it's
a
not good.
lenient teacher marking.
Wait, but I feel like the microscope damn hard to use, yeah. Like, wait, I feel like that's the hardest like concept I ever learned in my life. Like, I don't even know how to use the microscope.
I cannot help you.
Huh?
I
Why
can help you
cannot?
find video to help you learn how to use microscope. I don't have a microscope here, dude.
Oh,
I
no.
already said already, I only can help at most is like planning, okay, and like try to speculate. I'll be having a practical session soon. I can't remember when, but I think it's already included in the schedule.
Yeah, then
I think I was focused on that
I was
at
doing
that
the
time.
practical package that you last time give us. Then I do so many practice, then end up I get 4 out of 12, yeah. Yeah, I was like damn sad.
Then the rest of it,
Oh, the rest of it also damn bad, like,
can you
like,
see?
How should get you?
like damn bad, bro, like 20 something.
Can you let me see? Let me have a
no.
look.
Okay, okay.
Okay, once you have, then I have a look,
No,
then
I
I
see,
can
okay.
give you a better critique.
I feel like the planning, like I wrote like very well, but then I end up like doing so bad. But I
So
think
I have to
that-
see then I can give you a better feedback. You right now say already is quite opinionated still.
Oh,
Right? I need to see and assess reasonably first. Okay? Let me have a look.
okay.
Okay, once you're done, please let me mark. I'm just trying to showcase to you what are the keywords that you typically are expected to know. Any questions that I never get to go through, same thing, you'll be using, I will be giving out the answers, so no need to worry about that. Who here don't have the PNC PCR notes? Do you have PCR notes? Everybody have already? Okay. Let me just keep aside. Once you're done, let me mark. I want to see the way your craft answers. Always tell yourself for explain question, is it a keyword technique or an overview point technique? Part D. Your question is 2 what?
2A, B, and D.
Okay, let's see D. So status of your discharge points on mitotic cycle closely regulated. Explain the need to regulate the mitotic cycle tightly. Okay, so the question is, do you use the overview point technique and the keyword technique? Overview point technique, right? There's no keyword that you can see how the 3 marks are delegated, right? So in the overview point technique, what's the tip I gave you for the first sentence for the overview point? Answer directly to the question, right? Agree? So how come they are saying that the need to regulate mitotic cell cycle tightly is the mitotic cell cycle is controlled by what? Checkpoints that will help to what?
Prevent premature progression
Of the?
cell cycle.
Okay, so that will be maybe your first overview point, right? Subsequent points is an elaboration from that, right? So how come, how many marks would this be worth?
1.
I mean like 3 marks, what? So your first point is already a critical point, right? Then subsequent point is just 2 marks, right? So how many marks is this? 2 marks, right? So how do I delegate the 2 marks? It's because, number 1, you can tell me why the need to actually prevent premature progression of cell cycle, right? Second mark is what happens if you have dysregulation of your checkpoints. This is an elaboration from the overview point. You understand? So again, how come you must actually meet the requirements for each checkpoint? So that you know that.
Prevent,
I know, but also,
provide sufficient time.
yeah, this also will enable sufficient time, okay, for your this cell machinery to repair the DNA should there be any damage, right? So we also ensure, or you can also write in a phrase that every time we ensure every progression, there is always no DNA damage, alright? And then what happens if we actually do not actually have checkpoints? What can we further elaborate on? Is that, sorry?
Mutation.
So you think about this, if I have dysregulation of your these checkpoints, what do they lead to? What's the consequence for that? Don't just tell me mutation. What's the consequence if you have dysregulation of checkpoints? You will have, that means you don't have your G1, G2M you will have. What's the way, the effect? That is the keyword. Okay, whoever is done, let me mark. Oh crap. Sorry, Isabel. Okay, thank you. The 4 reasons gene mutation will not always lead to cancer. You say substitution mutation. Gene, so you leave silent mutation. So again, same amino acid. Oh, okay. mRNA codon codes for the same amino acid. This one I'll give you credit here. Next one, very good. Just need to state down 4 reasons. 1 more H, don't need to elaborate too much already. As long as you tell me degeneracy of the genetic code, so you code for the same amino acid, I'll give you the credit.
Okay.
Next one, you say DNA is double-stranded cell repair mechanism, can use other strand as a template to repair the mutated DNA. No, the idea is that again we already have mutation already, right?
Yeah, so you can repair the mutation.
You want to repair the usually this is not a point that we are looking out for DNA to be double-stranded for DNA repair, because the mutation already occurred. DNA repair, right, is when it occurs before the mutation becomes permanent. So usually we cannot accept this point. Okay, so you can accept the other points that just now I mentioned in class, which is a missense mutation.
Yeah, I wrote that.
So you have the one.
But then we need like 4, right? Then
Yeah, so let me explain step by step. You say can result in consecutive missense mutation.
Yeah.
Change in amino acid code for derivative acid with a similar R group property. So I'll give you the credit here. Then you have 3D confirmation of this is unaffected. Yeah. What's the other one?
I think I will initiate hypothesis.
No. So other mutation, and like I said, the missense mutation, that is also going to be a non-essential amino acid.
But is it not like the same as all?
So that's why-
mutation?
I know, but one is a different reason. One you replace with similar R group property. One you replace even though you change R group, but it's a non-essential amino acid.
Oh, okay.
So they will not lead to cancer formation. But another one could be what? Because this is actually a cancer formation, I also can consider, let's say mutation, right, happens in a tumor suppressor gene. But usually, to actually have loss of function in tumor
Wait, I
suppressor
was
gene,
gonna write that, right? But doesn't that technically lead to cancer? Because if you have mutation in one copy, then you're leading towards cancer. Because once you have a mutation in the other copy, then you
so,
get
but
cancer.
that is what could be one of the reasons may not lead to cancer, because maybe the mutations they occur in only one single copy of the tumor suppressor
But that's
gene.
like part of the step towards cancer. Right?
I know lah, but haven't yet lah. Can be tumor but not cancer.
Okay.
So remember I said tumor and cancer are different things.
So I cannot write like the initiate hypothesis.
Initial apoptosis, I feel like then if you can DNA repair can initiate apoptosis, but how the mutation in the, they are asking why
The
if mutation already
yeah.
occur, why still cannot lead to
Yeah,
cancer?
because if you initiate apoptosis, then you cannot accumulate.
But the,
mutation.
what do you mean? You listen up. They
Yeah.
are asking right why a gene mutation
Yeah.
may not lead to cancer. So let's say the question, right, or the examiners when they set this question, how
Yeah.
come they specify again gene mutation, the mutation, and that means you change your nucleotide, how come still doesn't lead to cancer? They are not asking you what are the fundamental effects that will still prevent cancer. If not, the teachers will paraphrase their sentence and say that why mutation may not lead to cancer. Why must they specify the word gene mutation? So I already said before in class, every single phrase that teachers come up with or set questions, right, is with intent one, it's deliberate one.
Okay.
So that's why by right, even though if I change this, if I want to accept your answer, I will change this to why mutation may not always lead to cancer. Not necessarily must be gene mutation.
Okay.
You understand?
Yeah.
So other things, however, just now I said the one copy, right,
Yeah.
of the tumor suppressor genes. And then you must, to me, you must also write in conjunction is that not only we must have both copies of the tumor suppressor genes that must be mutated, okay, but we also must have accumulation of multiple mutations within a single cell
The
lineage.
properties prevent the accumulation of mutations.
During preventing accumulation of
Yeah.
mutation. For this part, right, if in the event you say gene mutation, I think that the idea should be more of the accumulation of mutation in a single cell lineage is rare or by chance.
Okay.
Then that one will be a possibility. Rather than the perspective is very different. So accumulation of many gene mutation within a single cell lineage is by chance or rare. And that's why it will actually may not lead to cancer. Rather than the way you phrase prevent apoptosis, so you will not cause the accumulation of mutation.
Okay, yeah.
There's a different thing,
Yeah,
right?
yeah.
So that's why I will not accept. Okay?
Okay.
The other one. Evaluate the evidence. So evaluate, must you give a stand?
No.
So usually evaluate is not necessary must give a stand. You just need to see from the data whether it supports or does not support the hypothesis.
Yeah, I always don't know. Do I need both-sided?
So when do you have both-sided? Discuss.
Okay.
So when you see the command word called discuss then both-sided, comment, evaluate, need to take a stand.
Need to take a step.
Yes.
Also,
That means you need to show me how they actually
Oh, I
support.
oops.
Or you tell me how they do not support lah. I will accept both.
Oh, 'cause
You
I
know
wrote one support, one never support.
Oh no. So you notice how? Key thing is you see using the data given, right?
Yeah.
So it means that you definitely need code data, which also means that 1 mark is entails to quotation of data. So where is your specific quotation of data?
I think I have the percent.
The 99% survival.
Yeah.
Hey, your handwriting
Yeah, because
Lower.
probably got bigger space.
lower. It's not about the space right now. I'm looking at the way you write for this, okay? So again, with low gene expression of C, DK entry, and 99% after 1 year. Okay. So idea is that when you say support, equal does not always equal causation. Okay, so the idea is this, I accept does not always equals to causation because we see the quotation thing. Fine, I accept your answer.
Oh, so it's okay.
Fine.
Okay.
Okay, so because the idea is that there is insufficient data to say it,
Okay.
right? Okay, good that you can recognize proton oncogene that undergo keyword gain of function mutation become oncogene. Yes, always must write proton oncogene become oncogene. Yes, you say production of hyperactive product overproduction of this. I accept your answer. So your keyword, however, no, I will not see overstimulation of cell division.
Okay.
I must see uncontrollable cell division.
Oh.
So you have the one that I underlined, that overproduction of protein, but the keyword you're missing is uncontrollable.
Okay.
There's a difference, because sometimes you overstimulation, but you're still a normal cell, not a cancer cell.
Okay.
So that's why the phrasing really matters. Okay, who else? Can I mark?
I sent to you my
Oh.
answers.
You try the other question first. I'm marking the rest first. Give me a moment. 9 triplets of microtubules arranged in a ring. So you say how row, you say how centrioles help to organize mitoic spindle fibers. So that's why idea is that you organize mitoic spindle fibers during the assembly of mist. Please always write to produce microtubules. So not only they organize but also produce. Okay? So you organize mitoic spindle fibers and also produce. You synthesize the spindle fibers as well. But if you want to organize, the next key phrase, not just say produce, you tell me how to separate sister chromatids. We always use this phrase. Why do we need microtubules? Idea is to separate sister chromatids. So without the word separation of sister chromatids, I can't give you the credit. Okay? I don't care about you just merely telling me organize. Must tell me which one. Next one. Region occupied by a pair of centrioles. Centromere, why is it a region occupied by a centriole? Are you thinking about centrosome? But they're asking for centromere. So you're not using the keyword, they associate with what? Think about that. So is it a non-coding DNA of AT-rich region? Yes, right? And it also has complementary in shape to binding of your kinetochore proteins, things like that. So you can actually mention that. Okay, so function, you will say that associate with kinetochore protein to ensure proper alignment and equal separation of sister chromatids to opposite poles of the cell. Now, Yunchin, I want to tell you something. In exam, please try not to write like that. Really, you know, because marking is a very subjective thing. Teacher, I tell you, they mark thousands of paper. Alright, so you try to process your thoughts before you write down. Okay, but anyway, how you regulate my tightly. So you should know you're using the overview point technique. So this one is all irrelevant so far. Remember I say for overview point technique, what's the key thing that I gave you a tip for? Straight away answer the question. So why do you need to regulate tightly? Because the checkpoints, number one, prevent of the cell cycle. So always answer directly, okay? The checkpoints prevent that, and then you will tell me how come the remaining 2 marks is always to elaborate with effort, right? So how do we delegate the 2 even numbers? Number one, why must you prevent premature cycle progression of the cell cycle? Think about this DNA repair, provide enough time, etc. Right? And number two, what happens if you never regulate? You understand? So you mentioned about, again, prevent no genetic variation. Why do I care about no genetic variation? It doesn't answer to the question. And then you say prevent any mutation for abnormal. The idea, right, is not just prevent any mutation. So the idea you must tell me is that you want to make sure, actually, fine, I'll accept your prevent mutation. Fine. But this is not very good in the way you write. Later I'll comment. Who else is done?
Wait. The last one.
Isabel, I marked the last one because it's online. Give me a moment sorry. Degeneracy or genetic code. Why do you only emphasize about POC and TSG? Please don't need to emphasize about POC and TSG. Because you know telomerase must also mutate, right? Things and other cancer critical genes.
So I just don't talk about it. I don't mention like what genes.
Yeah, so can I ask, the moment I said that you want to talk about silent mutation and degeneracy and genetic code, we always have another key phrase that is always in conjunction with this. So you must always say the same.
Amino acid.
It's still coded for. So that is always the relevant keywords, right? So that's one thing I need to emphasize, because I right now have a feeling the way you craft is sometimes you miss out the key phrase that is always tagged along and associated with the word. So you need to really start to revise on paper too. A tip, people, is that if you want to familiarize yourself what are actually the keywords that is always associated with certain terms, spam or just study this paper 2 answers. Okay, because paper 2 is your revision material. May not be in the critical region. Now, again, alright. Critical region of what? Of your protein, is it?
But it's gene, then I don't know.
So, in other words, the amino acid, you will say itself, what's the keyword? It's a non-essential amino acid. That is the keyword tagged to it. You understand? Yes, may not be in a critical region, but you still need to elaborate. Just by writing this, right, it's very vague.
Okay.
Loss of function mutation could occur only in one. Huh? Really? Are you sure only can occur in one? Must always occur in 2. It's a recessive kind of mutation.
No, I was gonna say like because loss of function mutation needs.
Then you use the word loss of function is the wrong terminology already. Because loss of function, that means you no more function. So you mutation in just one. Do you see the phrase? So phrasing really matters here. Mutation in just one allele will not develop because the tumor suppressor genes, okay, is still functional. Am I right? So the fourth one can be what? For example, you think about similar R groups. It's a very different property for non-essential amino acid. Missense mutation that actually replaces the amino acid with similar R groups, so you have same conformational shape. Again, take note about that. Okay? So I can't give you any credit. Next one. Evaluate means you need to come out and stand with a supported evidence. So you say significantly lower significantly lower at for young upper compared to 99 and this low expression. High expression, low expression. So overexpression results in increased death, right? Fine. So if this is your quotation or data, what do we say? Evidence supports. Fine. I will accept that evidence actually supports the hypothesis. Oncogene transform into. Hey, you're phrasing again. Oncogene, is it the cancer one? Or proto-oncogene is the cancer one? Oncogene. So what do you always say? Gain or what do I always say? Gain or function? Mutation of proto-oncogene to become? So you write transform is also wrong terminology. Just train yourself. Transform is only for what? Bacteria. Okay? So I cannot accept if you say stimulate cell division. Because the keyword here, you want to become oncogene. You cannot just have cell division. Normal cell also can have cell division. What is the cell division do you want to describe? Yes. So because you are missing this key term, I cannot give you the credit either. Okay? Jude, are you done?
Yeah.
Can I mark? Alright, give me a moment, people. They are arranged in 9 triplets of. Wait!
Oh.
9 triplets of microtubules arranged in a
Cylinder.
ring.
Rod. Ring.
Okay?
Really?
Yes, really. Hello, really.
I asked you copy, you say you don't want.
See?
Right.
See?
I
Now
just
scolded.
said that. I
Kena
just said
scolded
that.
already. See? Kena scolded. So 9 triplets of microtubules
In a ring.
arranged in a ring. What's the second one? A pair of
Rod like.
structures
Yeah. Cylinder. 90 degrees.
to
Each
each.
other.
Yes, perpendicular each other. You don't shish. You see, you say you study a lot, but you're not writing the keywords. So, for example, I give you my 2 cents for that. Number 1, you're not writing keywords. Number 2, you're not answering directly to the context of the question.
You mean like
So
that?
maybe that's why
Like this
the
one?
practice paper when I was marking.
Oh, but this one like super duper hard.
No, it's not super duper hard. It's you are not recognizing the keywords, okay? That's why. So take note of the comments that I write at the side.
So messed up, yeah. 0 mark. Bro.
Yes, bro. So again, spindle fiber shortened. Why must you mention about that? So what are the features of having 9 microtubules arranged in a ring? What does your this centrioles do? Centrioles, right, do not attach to your this kinetochore spindle fibers. I think you have some misunderstanding. Centrioles are the one that synthesizes or produce spindle fibers. So they either organize your this mitoic spindle apparatus or complex so that you can actually ensure
Bind.
the separation of your sister chromatids, equal separation of sister chromatids, opposite poles of cell.
Separation.
Then the other one is the what? Also to align, right? So equal separation is for the other one, but this one, shouldn't you say the word equally? Now, you use the wrong terminology because you say they are attached to kinetochore spindle fibers. They do not attach to kinetochore spindle fibers. They actually organize the mitoic spindle fibers. Then you can say, which will help separate sister chromatids equally. So your phrasing here, this one is wonky. I cannot give you the mark here.
Wait, they all not attached together.
No, the other one make what? What do you mean attach?
I thought like make and attach like.
You say attach kinetochore spindle fibers. The centrioles are the one that that produces the spindle
Then
fibers.
it's like it's with the chromosome, right? Then it's like a string. Then they like pull it on the string,
But it's not a string one, right?
But then so the
I know the phrasing. I already tell you already. The angmo must be on flick. Your angmo is what is it called? Your Gen Z term or hayo. Okay, so take note. Proper alignment of chromosomes along the equator. Equal separation of sister chromatids are accept. Now I cannot accept the other one, which is the non-coding region, because I'm very strict. I need you to actually tell me it's made up a non-coding region of tandem repeats, or you will say a non-coding region that is complementary in shape too.